So now we get into the actual DNA replication process. You will recall from our biochemistry that the DNA is made up of nucleotides, and the nucleotides are numbered, right? If we actually look at the chemical structure of a nucleotide, we have a base, a sugar, and a phosphate group. And the actual nucleotide is numbered. We can number them. So I'll number it here on the screens. So you're with me. So this is carbon number 1 where the base is attached. Carbon number 2. Carbon number 3 is where the hydroxyl group is, right? Deoxyribonucleic acid. There's an oxygen group here. Carbon number 4 and carbon number 5. And carbon number 5 is where the phosphate group is attached. So whenever nucleotides are built into or polymerized into a nucleic acid, it always bonds in a certain direction. The 5' carbon always bonds to the 3' carbon. So that phosphate makes a bond, creating this bond between this nucleotide and this nucleotide. So carbon number 5 bonds to carbon number 3. That's important. And for that phosphate to bond, there must be an oxygen. That oxygen has to be there. If there is no oxygen there, then this new nucleotide can't bond. So that bonds to that oxygen, and you end up building a nucleotide into a nucleic acid. So that's very important. So whenever we refer to a nucleic acid, we always read it from a 5 to 3 direction, 5 to 3. So I'd read this as ATGCCC. If 5 were on this side, on the opposite side of the screen, if the 5 were over here, and the 3 were over here, then I'd read it CCCGTA. You always, always, always read it from a 5 to 3, no matter where the 5 is, no matter where the 3 is. Okay? So when I look at my DNA double helix, I have this strand that goes from 5 to 3 on one side and 5 to 3 on the other side. They're anti-parallel. They're going in opposite directions. And these two strands are not covalently bound to each other. They're actually held together by hydrogen bonds, which is important because the DNA, in order to replicate, this has to be able to be separated. And if you have a strong covalent bond, you need to input an enormous amount of energy to separate the two strands. With a hydrogen bond, in great numbers they're pretty strong, but individually they're relatively easy to break. So the hydrogen bond is a perfect bond that will allow, when broken, will allow the two strands to be separated. Okay? So, if you follow along with the PowerPoint, and I might put the PowerPoint up in a corner so we can kind of follow along with it together, I'm going to be referring to the PowerPoint as I go through this entire model here. So this model here represents the replication fork. It is all the things necessary for DNA to replicate. You'll see that there are a lot of players here. There's a DNA, of course, and there is this big, giant, massive DNA polymerase. And there's a few other things which I'll mention as I get to them. Now, if you look at my PowerPoint, you'll see that I've talked about the 5 to 3 direction on the PowerPoint. And then when I get to step 1 on slide 4, this is the DNA replication process. So initially, your DNA is wound in a helix. Right? So it's a helix that looks like this. And if I were in class, I'd be able to show you the helix as a model, and I could show you how the helix would have to be unwound. So you need to get access to the nitrogen bases. In order to get access to the nitrogen bases, you have to unwind the helix. And the way it's unwound is thanks to an enzyme known as gyrase. So the gyrase comes along and it nicks the backbone, right? It nicks the backbone a little bit, and what it does is the DNA is coiled. I don't know if you can see my hands, but it's coiled, it's twisted. And so it will nick the backbone, it will allow it to untwist, it will introduce a negative coil, so it will actually untwist and go in the opposite direction. So it's no longer twisted. So that's gyrase's job. And gyrase is a particular class of enzyme known as a topoisomerase. But gyrase is what's important. So it does this job, moves on. Next, we have to separate your DNA double helix. We've taken out the tension of that helix. It's now able to be split apart. So you need something that's going to separate the complementary nitrogen bases. And so that special enzyme that's going to do that, they're called helicases. So along comes a helicase. I've already split apart this DNA slightly to help me with my model, but the helicase is going to come along and it's going to split the hydrogen bonds. So if I had this on a different screen, it would be my eraser. And it would cut through all these, separating the hydrogen bonds, and then it allows the DNA double helix to expose the nitrogen bases, because that's what you need to copy. Next, step 3. This C, and you can't really see it there, this C and this G and this C, this A and this T, they're all attracted to each other. So just because I separate them with the helicase and it's done its job and now it's gone, doesn't mean that these won't come back together again. So you need something to stabilize this structure, to keep it open so they can be copied. And so that's the job of these SSBPs, which stands for single-stranded binding protein. So you got a group of these that will bind to the DNA, and think of them as like light-charged magnets or something, but they keep the strands separated. And so now the fork is exposed. That's this area here. I keep on calling it a fork. And you can now get replication. Now, we are ready to replicate. So if I were to ask the question in class, the question would be what do you think the next enzyme is that's going to replicate the DNA? And it seems to make sense that that next enzyme would be DNA polymerase. Here it is, a big giant DNA polymerase. It's its job to polymerize DNA, to make copies of the DNA. However, DNA polymerase requires 2 conditions. Condition number 1 that it requires is it must have a free DNA template, and it must have a free 3' hydroxyl group. So let's take a look at what we have here. Is there a DNA template? Yes, there is. This thing right here represents a DNA template. So there is a DNA template, that's fine. Is there a free 3' end? Right? Is there a free 3' end? So when I take a look at this DNA, we want to make a brand new DNA right here. So for me to make a brand new DNA with all the little nitrogen bases, all the nucleotides that are being added, I need a free 3' end right here. And as you can see, there is none. So because there's no free 3' end, the DNA polymerase can't start, it can't latch on. So we need something else. We need something to start the DNA polymerase process. We need something to prime it. And what are we going to use to prime the DNA replication? We're going to use a primer. A primer is a single-stranded RNA, a short sequence. In order to build a primer, you need a special enzyme known as primase. So the primase will come at the very beginning, and it only needs one condition to work. It only needs a template. It's got a template. It doesn't need the free 3' end. So the primer will come along, the primase will come along, it will create a primer. I'm making it super short for my model here. A primer is a little bit longer than this. Primase adds the primer, it's done its job, off it goes. So now that there is a primer, an RNA primer, so that's kind of weird. It's not DNA. We want a DNA helix created, so this is something we have to address later. Now that we have this RNA primer, let's see if our conditions are met. Do we have a template? Yes, we do. Do we have a free 3' end? Well, my DNA strand on this side, it's 5 on this side. I have to tell you that. It's 5 on this side, and it's 3 on this side. The new nucleic acid that's being built on it would be 5 over here, and it would be 3 on this side. So yes, there is now a free 3' end right with this U. So that means the DNA polymerase is now able to do what it's supposed to do, and that is polymerize the DNA. Notice how I focused on one strand. Right now I'm focusing on the top strand because this top strand makes sense. In the top strand, which is running in a 5 to 3 left to right direction, the new strand that's going to be created is going to go from a 5 to 3 in a right to left direction, which is fine because my DNA polymerase is moving from the right side to the left side. So as it comes along, this big giant DNA polymerase, notice it's number 3. It's going to find the C, and it's going to find the complementary nucleotide and add a G. There's another C beside it, so it's going to find another nucleotide that's just floating around the nucleoplasm and add it. To this G, you're going to add a C. And to this T, you're going to add an A. To that G, you're going to add a C, and so on. I'll just do one more because after that I run out of room. So this DNA polymerase is moving along, and as it moves along, it's able to polymerize this strand. And when it's added, what's actually happening is they're being covalently bound, right? So these are actually all covalently bound to each other as you would expect for a nucleotide, for a nucleic acid. And this keeps on moving along, and as this strand keeps on separating, you're going to end up getting a DNA double helix from the top strand. It'll be perfect apart from that weird RNA molecule at the end. So here's where the problem arises. The DNA polymerase moves in one direction, but the bottom strand, because it is anti-parallel to the top strand, so the top strand runs from a 5 to 3 from left to right. That means the original bottom strand runs in a 5 to 3 from right to left. It means that any new DNA created on here, which I'll use blue to represent it, right? Oops, that's not blue. Has to be created in this direction, right? Because the 5' end will be over here, right? Anti-parallel. And the 3' end, I have a 3 on here somewhere. Ooh. I'll just grab this one. Will be over here. And remember what I said, when DNA is created, it must be created in a 5 to 3 direction. If this DNA polymerase is going in this way, and that means there's a 5' over here, it doesn't make sense. It's backwards. It can't work like that. So the bottom strand ends up being a problem. The bottom strand is actually, to fix that problem, to get past that problem, it's created in little segments. So the first thing for the DNA polymerase to work on the bottom strand, you need a template, which we have. I'll keep on circling it. We have the template. That's fine. We also need a free 3' end, which we don't have. So that means the primer, a primase, has to come and produce a primer on the bottom strand as well. But it's got to do it on the left-hand side now. So I'll just create this primer using red as opposed to using my letters, and I'll just create a little primer. So my primase has created a primer here, and now it moves off, it's gone. This is my 5'. I know it's getting really messy, that's why I have to follow along. This is my 3'. And now the DNA polymerase for this short little section is able to create a little tiny segment of DNA. So to that A, it will bond to T. To that C, it will bond to G. To that G, it will bond to C. To the next G, to the next G will bond to C, which I don't have any more of, and to the next T will bond an A, and so on. So it will just produce a small little fragment. Clone. No, clone. And let's clone this one. Clone. To that T, we'll get a G. And then finally, there we go. So now, on the top strand, remember the top strand will be continuous. The top strand is perfectly fine. Let me just go back to my model over here. It'll be a little bit easier for you to follow. So when I take a look at my top strand, those separate. Get rid of those. So to take a look at my top strand, when I form my new strand, which I'm doing in blue, it's formed like this. It's perfectly fine. So we end up with a good double helix. But on the bottom strand, you need a primer, and then you end up with this little fragment that's produced. And then as the DNA polymerase keeps on moving, as it keeps on moving along here, right, it's going to produce another short little fragment. So there's another primer and another piece of DNA. And it's going to keep on moving. So this top strand is created continuously, which is not a problem. But the bottom strand is created in little tiny fragments with lots and lots of these primers. And so that's how the cell gets around the problem of DNA only be creating in one direction. So the top one is a continuous synthesis on the top strand. The top strand is called the leading strand and it has continuous synthesis. The bottom strand is called the lagging strand. It happens at the same speed, but the overall speed of the bottom strand is slower, slowing down the whole process. So this is known as discontinuous synthesis because it's made up in fragments, little fragments. All right, so the DNA polymerase moves along, and at the end of this, you'll end up with 2 full strands. But it's not DNA double helixes yet because you got all these little chunks of RNA inside of them. So the RNA has to be removed. In order to move the RNA, we have DNA polymerase 1. So the DNA polymerase 1 will come along, and what I'll do is I'll just grab it from here, and I'll go back to this other page where it's not as busy. The DNA polymerase 1, it will remove the primer. So let's get rid of that primer. Gone. Delete. And it'll get rid of that primer. Delete. And then the DNA polymerase will, like any other DNA polymerase, it needs 2 things for it to work. 1, a template. Is there a template? Yes, there is. Template's right here. And it needs a free 3' end. Oops. So does it have a free 3' end? Yes, it does. Right? The free 3' end is now sitting right here. So now that it has both those conditions met, this DNA polymerase, once it removes the primer, it can then fill in the hole. So it inserts the gap with new DNA. But notice when I inserted it, it's not attached, right? It's freely movable. It didn't covalently bond them. So you still need one more step here. That one more step is you need to glue it all together. So there's one more enzyme known as ligase. And what the ligase will do is it will come along and it will glue, basically create a covalent bond between the newly inserted nucleotides and the original nucleotides that were created from the polymerization process. So the ligase is a glue. It's a DNA glue. So it sticks it all together. That's the ligase's job. Right? So now all those little primers have been removed and we filled the hole, and are we done? Well, you'll notice that there's still something a little weird happening here. And the weird thing is this. There is this little primer at the end. And when these primers at the end get removed, the DNA polymerase is not able to do what it needs to do. So let's say this primer is gone, right? The DNA polymerase removed the primer, but is it able to fill that hole? Does it have a template? Yes, it does. Does it have a free 3' end? Well, there's a 5' end here, but there's no free 3' end over here. That means this DNA polymerase cannot fill that gap. Because it can't fill that gap, it means that the DNA will be shorter. every single time the DNA replicates, it gets shorter and shorter and shorter. And so that becomes problematic, because if your DNA gets too short, then you start wearing away your genes. And if your gene's coding for some very important cellular process, then that cellular process won't happen. So the way that your body's able to deal with that situation is that in utero, when you're very young and your cells in your body are very young, you have a special enzyme. And that special enzyme is known as telomerase. So I don't have any here. I thought I had a model here, but so what the telomerase does is it creates these telomeres, and telomeres are just long fragments of, we'll call it junk DNA, that's just attached to the end. And so the telomeres, they get shorter and shorter, but it's okay. With every single replication, your telomeres get shorter, but the telomeres don't represent anything, right? They're just junk DNA. So with each replication, the telomeres get shorter and shorter and shorter and shorter. But then eventually your telomeres are gone. And if your telomeres disappear, then your genes on your DNA will be affected. This is one of the reasons why we have a maximum lifespan. Your cells have a maximum number of times that they can undergo replication. And once you've hit that number, that's normally associated with the telomeres being completely expired. This is something that helps define something called the Hayflick limit, which we talked about in grade 10. There's a lot of scientists that believe that the telomere, if we can extend it, then we could extend, that could be one of the key ingredients to extending our lifespan. Because as the telomeres are gone and the genes degrade, then other processes degrade as well. There are also some cancers that are actually able to activate telomeres and create a infinitely lived cell, which is what cancers are. They basically live forever. In our class, I don't have time to talk about the HeLa cells, but this is a fascinating area, and it's one of the things that I might get you to read as an interest above and beyond the content. But at this point, we have explored the DNA replication process. So you should go through the PowerPoint, make sure you write down the notes, review this video if you need to. I'll put up a link to some other videos that look nicer, and you hopefully will have a good understanding of how DNA replicates.